Welcome to the Genetic Code Room. Here I will enlighten you with the coding dictionary and the characteristics of code. We will begin with the coding dictionary. The techniques used to decipher the genetic code have left us with the coding dictionary of 61 triplet codon amino acid assignments. The remaining 3 triplets, UAA, UAG, and UGA, are termination signals, not specifying any amino acid. The code is degenerate, meaning all amino acids are specified by 2, 3, or 4 different codons. 3 amino acids, serine, arginine, and leucine, are each encoded by 6 different codons. Only tryptophan and methionine are encoded by single codons. Another evident pattern is degeneracy. Most often in a set of codons specifying the same amino acid, the first 2 letters are the same, with the third letter differing. Francis Crick observed a pattern in the degeneracy at the 3rd position, and in 1966 he postulated the wobble hypothesis. This first predicted that the initial 2 ribonucleotides of triplet codes are more critical than the third member in attracting the correct tRNA. He thought that hydrogen bonding at the 3rd position of the codon-anticodon interaction is less constrained and need not stick as specifically to the established base pairing rules. The wobble hypothesis proposes a more flexible set of base pairing rules at the 3rd position of the codon. This relaxed base pairing requirement allows the anticodon of a single form of tRNA to pair with more than one triplet in mRNA. With the wobble hypothesis, U at the 1st position of the tRNA anticodon may pair with A or G at the 3rd position of the mRNA codon, and G may likewise pair with U or C. Inosine, a modified base found in tRNA, may pair with C, U, or A. Applying these wobble rules, a minimum of about 30 different tRNA species is necessary to accommodate the 61 triplets specifying an amino acid. Wobble can be considered a potential economy measure, provided that the fidelity of translation is not compromised. Current estimates are that 30 to 40 tRNA species are present in bacteria, and up to 50 tRNA species are present in animal and plant cells. How to use the Genetic Coding Dictionary using the example codon AUG. First, look to the left-hand side of the dictionary, first base. The first base in AUG is A. Next, look to the second base. The second base in AUG is U. The third step, look to the third base. The third and final base in the codon is G. Where the highlighted portions meet up, you will find the amino acid which correlates with the codon AUG. This amino acid is MET, or methionine. Now we will go further into the history of the coding dictionary and how it came to be. We already examined Francis Crick and the wobble hypothesis. Another scientist, Sidney Brenner, argued that the code must be a triplet. 1 or 2 letter words would not allow for the coding of 20 amino acids. With 3 letter words, we can code for 64 different amino acids, enough for our 20 and then some. Brenner also argued that the code is non-overlapping. The number of tripeptide sequences should be limited with an overlapping code, but Brenner found that it was not. Now on to Marshall Nierenberg and Philip Letter. The Nierenberg and Letter experiment explained the triplet nature of the genetic code and allowed the remaining ambiguous codons in the genetic code to be deciphered. In this experiment, using ribosome binding, various combinations of mRNA were passed through a filter which contained ribosomes. Unique triplets promoted the binding of specific tRNAs to the ribosome. By associating the tRNA with its specific amino acid, it was possible to determine the triplet mRNA sequence that coded for each amino acid. Nierenberg also developed a system for synthesizing proteins in vitro with J.H. Mathai. They had learned that preparation of disrupted cells soon ceased to make protein, and, in an attempt to prolong the short period during which in vitro synthesis continued, they added RNA to the preparations. These two explain the nature of a codon. Another contributor to the genetic code is Hargobind Khorana. He is given credit for cracking the genetic code. He devised biochemical methods to produce nucleic acids. The first one he made was a strand repeating the two nucleotides UCU-CUC. This translated into a strand of amino acids, reading serine-leucine-serine-leucine. Synthetic RNA were later used to decipher the rest of the genetic code. Now we will move on to the 8 characteristics of genetic code. 1. The genetic code is written in linear form, using the ribonucleotide bases that compose mRNA molecules as letters. The ribonucleotide sequence is derived from the complementary nucleotide bases in DNA. Each word within the mRNA contains 3 ribonucleotide letters. Each group of 3 ribonucleotides, called a codon, specifies 1 amino acid. Thus, the code is a triplet. 3. The code is unambiguous, meaning that each triplet specifies only a single amino acid. 4. The code is degenerate, meaning that a given amino acid can be specified by more than 1 triplet codon. This is the case for 18 of the 20 amino acids. 5. The code contains start and stop signals, certain triplets that are necessary to initiate and to terminate translation. 6. No internal punctuation is used in the code. Thus, the code is said to be commaless. Once translation of mRNA begins, the codons are read one after the other, with no breaks between them. 7. The code is non-overlapping. After translation commences, any single ribonucleotide at a specific location within the mRNA is part of only 1 triplet. 8. The code is nearly universal. With only minor exceptions, a single coding dictionary is used by almost all viruses, prokaryotes, and eukaryotes. Thanks for visiting the Genetic Code Room. You will now move on to the Transcription Room.